आज सुबह 8:30 बजे आ गए लगा नायका से पार्सल।
Good evening, sir.
Good evening, everyone.
Good evening, sir.
Yeah,
Good
good evening,
evening.
Jitendra. Jitendra, good evening. Okay, so in the last class, we had started with atoms and molecules, right?
Yes, sir.
Okay, and we had seen the laws of chemical combination. And before that, I don't think that laws of chemical combination is coming for anyone's exam. Is it coming for your exams? Laws of chemical combination. Is that chapter coming for your exam? Laws of chemical combination, I don't think it's coming
Sir,
for
let me
everyone.
check. Sir, it is not coming.
Yeah, it's not coming. So that's what I think, that laws of chemical combination is not coming for your exams. Okay. So what I think that, however, we have started with that chapter, we can carry on with the revision of other chapters, like the chapters we have done in chemistry, the chapters we have done in physics, and today we can complete the chapter cell. Okay. We have got 3 classes, 1 hour each, from Okay, so we have got these three classes, and out of the three classes we have done one class physics, one class chemistry. So today we will be doing one class bio. So every day we'll be completing like this, only one class physics, one class chemistry, and one class biology. Okay, we have to do chapter cell. Okay, so in cell we had done the different parts. Like we were doing about the nucleus, we have done the differences between plant cell and animal cell also. Okay, so let's continue with the chapter cell. Just I want to carry on with the NCRT first. We will see what else is remaining from that chapter. So NCRT. Okay, is there anyone in who is also getting the chapter tissue?
Sir, me.
Okay, so I think just we finish up with this chapter cell. We have done almost everything from this chapter cell. Just I
My
need to.
exams are completed.
Your exams are from?
Completed, sir.
Okay. So see, I was also surprised to see some of the answers from this chapter cell. Like I gave you the question regarding osmosis. So I think you still have some doubts related to osmosis. And osmosis, plasmolysis, deplasmolysis, and tonicity of the solution. Okay. And some of you had, I think, also written the differences between prokaryotic and eukaryotic cell were incorrect. Okay. Also, we are left with the small part, that is cell division. So today we'll be completing these parts, whatever are left from this syllabus. So let's see. So I have a cell division or cell. So for cell, let's see cell division. So see, as we know that all cells arise from pre-existing cells, right? So all the cells, they must divide to regenerate or give birth to the new cells. So since from the cell theory we have all cells arise from pre-existing cells. So definitely the formation of the new cells, it will be carried on from the previous cells only. If you have any previous cells, so from then only there will be the formation of new cells. So here let us see how it happens. So we consider here any cell. Let's say we consider here an animal cell. And in the animal cell, we will be looking first of all inside the nucleus. So the cell division or the birth of a new cell from the pre-existing cell, it consists of different stages. So what are the different stages of which the cell division consists of? See, here you have cytoplasm, nucleus, and inside the nucleus you have the nucleus. In the nucleus you have chromosomes which are present, or chromatin fibers, and we know that the chromatin fibers are the ones which are the carrier of the hereditary information. So here you have the chromatin fibers which are the carrier of genetic information. So since the genetic information should be available with all the cells, okay, so whatever new cells are formed, they must carry the genetic information. So first of all, we consider the cell division arising from in two parts. So cell division we consider in two stages. Your first stage of the cell division, we consider the division of nucleus. So first of all, we consider here the division of nucleus. The division of nucleus is also called as karyokinesis. So first step is division of nucleus. So when the nucleus divides, all the nuclear material also divides. And the nuclear material divides in such a way that the genetic material which is present in one cell, it will be present in the all the cells which are generated. Suppose two cells are generated from this, which we will be calling as the daughter cells. So suppose one cell gives birth to two other cells which are called as the daughter cells. So the amount of the genetic material should be distributed equally, right? So both these cells, they should carry the same amount of the genetic material which are found from this. So first step is the division of nucleus. Then the second step is the division of cytoplasm. Okay. So division of nucleus, first step, that we also called as karyokinesis. Then the second one is division of cytoplasm, which we also call as cytokinesis. So cytokinesis means division of the cytoplasm. When the cytoplasm divides, then only it's cytokinesis. So first of all, the division will occur in the nucleus. Now this division or cell division is categorized under two categories. So we study cell division under two categories again. So what are the two categories of the cell division? See, the previously
Mitosis and meiosis.
yeah. So cell division is studied as mitosis and meiosis. In the mitosis cell division, it occurs in the case of somatic cells. All the somatic cells of the body, they undergo mitosis cell division, whereas meiosis cell division, it occurs in the germ cells. Now, what are the germ cells? The germ cells are the cells which are capable of forming the new organisms. Okay, so a single germ cell is capable of forming the complete organism. So those are the germ cells. For example, in the case of male, the germ cells are the sperms, and in the case of female, the germ cells are the ovule. Okay, so meiosis occurs in the
Yes,
germ
sir.
cells. Yeah, what happened?
Okay, sir.
Okay. So for the meiosis, it is found only in the case of germ cells. Mitosis occurs in case of somatic cells. So that is, it can be considered in all our body cells, like the cells of your skin, the cells of your heart. So all your body cells, they generally undergo mitosis cell division. And it is not possible to generate the complete organism from the cells of your body. Okay, a new organism can be generated only from the germ cells. Okay. So we'll first study the mitosis cell division. In the mitosis cell division, a single parent gives birth to two daughter cells. So here you have the formation of two daughter cells in the case of mitosis cell division, whereas in the case of meiosis cell division, you have the formation of four daughter cells. स्लो करके चला जाना, स्लो करके 3 पे 3 3 3 पे. Okay. So also, if you look for the chromosome number, so in the mitosis cell division, the chromosome number is constant or the chromosome number is maintained. So here, the chromosome number is constant. It is maintained throughout. Whereas in the meiosis cell division, it is also called as reductional division. So here the chromosome number reduces to half. Here, reduced to half. For example, in the human body, a total of 46 chromosomes are present, which we also call as 23 pair. So there are 23 pair of chromosomes, which we represent as 2n. So 2n means the chromosomes are occurring in pairs. So n is 23, and hence there are 23 pair of chromosomes present in human body, present in the human body. So during the germ cell formation, that is during the formation of ovum, ovum or during the formation of the sperm cells, there is formation of, or there you have the formation of only 23 chromosomes each. Okay. So when the germ cells are formed, each cell will have just 23 chromosomes and not 46. So eventually each cell is having 46 chromosomes, but when the meiosis cell division occur, that is the germ cells are formed, they have only 23 chromosomes. Whereas in the case of mitosis cell division, the chromosome number will be maintained. So if here the chromosome number is suppose for 46, so still even after mitosis cell division, the chromosome number will remain the same. It will remain 46 only. So now we'll see in detail how does mitosis cell division occur. So for today, we'll be studying in detail about the mitosis cell division. Got it? Okay, so let's see what is mitosis cell division.
Sir,
Yes,
sir, can you turn the page back?
back. Yeah, yeah.
So one second. Okay, sir.
Okay. So, let us see mitosis cell division in detail. So mitosis cell division, it is studied under different phases. There are different phases of mitosis cell division recorded as the changes that occur at each phase. Okay. So the different phases of the mitosis cell division, they are prophase, metaphase, then you have telophase, the next phase is your anaphase, okay, before telophase you have anaphase, then you have the telophase. So you have anaphase, then you have telophase. And after telophase starts the cytokinesis. So cytokinesis, we studied that the cell division. These are the phases. That means prophase, metaphase, anaphase, telophase. They are the phases of karyokinesis. That means the process of division of the nucleus. So during the process of karyokinesis,
Sir,
yes,
sir, my science exam is on 23rd on Tuesday. Can you give me a revision of the graphs, if you can, on motion?
okay, you want the revision from graph.
Yes, sir.
Okay, so what you can do, I think I have sent one of the assignment of the chapter motion in the group.
Yes, sir.
Is it there? Just check it if it's there.
Yeah, sir. You sent force and laws and motion also.
Okay, motion is also there. So just see that there must be some of the questions based on the graph.
Okay, sir.
Okay, so just after this class, around I can give around 15 to 20 minutes for that graph. Okay.
Yes, sir.
Okay. So see, now what happens, we'll try to study each of the phases, what happens with the each phase. So during the karyokinesis, that means during the cell division process, the first step being the prophase. So the prophase, it is characterized by thickening of, chromosomes. So this is the main characteristic by which we can identify that prophase has started. In the initial stages, when during the normal cycle of the cell, during the normal life cycle of the cell, the chromosomes, they are very thin. They are thread-like structures, which we also call as chromatin fibers. So you observe chromatin fibers present in the nucleus, which are thread-like structures. But as the cell starts to grow and it is prepared for the cell division, that is karyokinesis is about to occur. So before that, the chromosomes become thick in structures. Okay. So what was the earlier structure and what is the new structure here? The chromosomes earlier, they might be something like this. So this is, if this was your nucleus, so your chromosomes were just these thread-like structures. But after the prophase one, you will be able to prophase, you will be able to observe the chromosomes something like this. You will be able to observe them here, some structures like this. So these are your chromosomes. So see, if you identify this in this way, you will find a number of the chromosomes. So here these are the chromosomes. See, if we identify the different parts of these chromosomes over here, we call these as chromatids. So these structures which we see on both the sides, they are called as chromatids. Right? So these we call as chromatids, and for this one they are called as the sister chromatids, since they are lying on the same centromere. So sister chromatids are the chromatids which lie at the same centromere. So this, the central part to which the chromosomes are attached, this is your centromere. So we call it centromere. Okay. Now the genes or the DNA, they are located on these chromosomes. So genes are the segments of DNA which are located on these chromosomes. Okay. If any exchange of genetic material, there is a process here which we call as DNA copying. So here DNA copying occurs. DNA copying means the genetic material from one of the chromatids to the other chromatid is transferred. Okay, so definitely, first of all, it was a single chromosome. Then from this, you are forming these sister chromatids. So there will be DNA copying. A copy of the DNA is generated. So whatever was the basic DNA of the cell, a copy of that cell, a copy of that DNA is generated, and it is transferred to the secondary chromatid or to the other chromatid. Got it? So initially, if the cell was formed, so it just had a chromosome like this with the genetic material present on it. But during the development of the prophase one, it led to the formation of the sister chromatids. Now the genetic material should be present on both the chromosomes. So now both the chromosomes, they get the copy of the DNA by the process of DNA copying. So during this process, whatever the genes are present on one of the chromatids, they are also transferred to the second chromatid. Okay, and this ensures that the new copy of the cell or the new cell that we get, it has the same genetic material as the original one, or both the cells which will be obtained by the mitosis cell division, they will have the same genetic material. Got it, everyone? Any doubts here? Any doubts
No
with
doubt,
regards?
sir.
No, okay. So that's nice. Here, during the process of DNA copying, you obtain your two chromosomes. Then next step is metaphase. So you have metaphase, then anaphase, telophase. Okay. So your metaphase is characterized. Okay. So during metaphase, what happens? During metaphase, you have the chromosomes which are present over there. The chromosomes, they are attached to the spindle rays. So we call them as spindle fibers or spindle rays if these are animal cells. So in this animal cells, you will have the appearance of centrioles. And here we observe the role of the centriole in the cell division. So see how do the works. This stage is metaphase. Now see what happens during the metaphase. You will have your chromosomes like this. And if it is an animal cell, you see. Now if this is an animal cell, so you will have the presence of centrioles here, and there will be spindle rays which appear from this centriole. So these rays will appear from the centrioles and they will attach the chromosomes to themselves. Okay. So this phase is your metaphase. You will find the appearance of spindle rays from the centrioles. If it's a plant cell, so in the plant cell there will be appearance of spindle fibers, but there are no centrioles. So without the centrioles, there will be appearance of spindle fibers. These spindle fibers will pull apart the chromosomes. Okay, so these are also called as microtubules, or we also call them as spindle rays or spindle fiber. So they appear at the next stage, at the anaphase stage. The next stage is anaphase. So at the stage of anaphase, what happens? Okay. So at the stage of anaphase, the two sister chromatids, they are pulled apart by these microtubules or spindle rays. So the pulling apart here, your metaphase, it is characterized by appearance of spindle fibers. Appearance of spindle fibers. Then anaphase is pulling apart of the chromosome. So the chromosomes, they are pulled apart here. So the two sister chromatids, they get separated out and they start moving towards the respective poles. So this phase we call as anaphase. So in the case of anaphase, pulling apart of sister chromatids occurs. Pulling apart of sister chromatids. Okay, the next phase is telophase. During the telophase, the so during the telophase, what happens? During the telophase, the chromosomes or the chromatid fibers.
Sir, may I leave class 5 minutes early
Okay,
for studying for science exam, sir?
so that's what I wanted to. You are only asking for that part of graph, right?
Yes, sir. No, sir. Yeah, sir. I'll stay. I can stay for 15-20 minutes more.
Okay. So see, for the telophase, during the telophase, the chromosomes which have been pulled apart, they collect at the two poles and they coalesce to form their clusters. So again, the chromosome clusters, they are formed at each poles. So in the anaphase, the chromosomes, they were pulled apart, whereas in the telophase, they collect at the two poles, and now the division of the nucleus starts. So here you will have the appearance of a meniscus in the nucleus, so that which marks the division of nucleus now. So here now the division of nucleus also occurs, giving rise to two nuclei. So after the telophase, here there will be the formation of, during the telophase only, you will see the formation of two lighter nuclei. So there will be two lighter nuclei which will be formed in the same cell. Now, after this starts the cytokinesis. So, during the stage of cytokinesis, up to now it was only within the nucleus. So, now during the stage of cytokinesis, all the nuclear material has divided, nucleus has divided. Now, only the cytoplasm material has to be divided. So, it appears something like this. Now, the cytoplasm forms a meniscus over here, giving rise to with two nuclei each. Okay. So, further, two daughter cells are given birth by this process. Okay. Since during this process, whatever cells which are obtained over here, they are called as daughter nuclei. Okay, so these two daughter cells which are formed, they are also called as diploid cells. Diploid cells means the chromosome number is still diploid. It's still the same. Whereas during the meiosis, you have the formation of haploid cells. Okay, so just check all the steps. Jitendra, what are the different steps for this? Yeah, Jitendra, can you tell the different steps? Harshit?
Yes, sir.
So
Yes,
Harshit,
sir.
can you tell the names of all the phases?
So prophase,
Prophase,
metaphase,
metaphase, anaphase, telophase.
anaphase, telophase.
Okay, prophase, metaphase, anaphase, and telophase. Right. So, okay, let's see all the diagram, all these phases through a diagram. So I'll just show you, share a PDF with that with all these diagrams. Let's assure and see what are the different phases. Just 1 minute, I'm sharing the PDF. One minute. I just want some pictures related to this. Okay, so here this diagram gives you a complete idea of the different phases which are involved.
Correct. Yes, sir. Sir, are these mentioned in the textbook?
Yeah, these are.
Sir, if they give a question like this, how many marks will it come for?
So its detailed content is not there. It's not the detailed content.
No, sir, like if they give it in, if they give it as question, how many marks can it come for?
Nearly 3 marks.
Sir, it can't come for 5 marks, no?
No, see, actually, if you look up for your questions, so none of the questions in the board exams, they are for a direct 5 marks. Okay, so there is no direct question of 5 marks. That means you don't get a single question of 5 marks or 3. The questions are divided into parts and subparts. Okay, so any question, if you observe, the question is not for the same. This was the one which we did.
Yes, sir. Sir, can they give it? Sir, if they ask us to draw the diagram, it can be for 5 marks, no?
Just let me check. I think it should be.
Sir, because when we were discussing in school, our teacher didn't tell us about the anaphase and
Okay, your teacher didn't tell about that. She only told about the cell division, right?
Yes, sir, only cell division, no anaphase, telophase.
Okay, okay. So still, I think we should have a complete knowledge of the everything. Okay,
Yes, sir.
let's see. So I think this is the rationalized content that is there. So that's why up to now I also didn't took that part. Just when I observed your book. Okay, so that was the previous one. Not much details are given in the new one. Okay, just you need to be aware of the different phases that the cell is going for the cell division. So see
Sir,
here
can
what
you post this picture on the group if you can?
okay, this picture.
Yes, sir, for the phases.
Okay, I'll try to send it. Actually, it's in the PDF, so I need to take a screenshot of it. And instead, if I take a screenshot, rather you can take a screenshot of the same.
Okay.
See, in the first phase, your first phase was the pro phase, and during the pro phase, you have condensing and cooling of the chromosomes. So that means during the pro phase, you have the formation of the chromosomes. Before that, before the pro phase, you can observe that the chromosomes, they are thread-like structures. So here, The chromosomes, they are just thread-like structures. Here, the chromosomes are becoming thicker. Okay, so you have the appearance of chromosomes from the chromatin fibers. During the metaphase, you have the appearance of spindle rays. During the anaphase, there is pulling apart of the chromosomes. Your chromosomes, they are pulled apart towards the pole. During the telophase, you have again the condensation of the chromosomes into two nuclei. There is formation of two lighter nuclei during the telophase, and after that, the process of cytokinesis starts. That means now the cytoplasm material is divided into two parts, giving rise to two daughter cells.
Arvind,
Here,
sir.
yes,
Sir, is this similar to the binary fission which we learned last year?
see in the binary fission, yeah, this is the similar to your binary fission only. In the binary fission, you didn't go into the depth like how the chromosomes are getting divided.
Yes, sir.
Okay, you just studied that first the nucleus divides into two and then the cytoplasm divides into two.
Yes, sir.
Okay, so over there you are not worried how the division is occurring inside the nucleus. So this is a general mitosis is a general cell division which is found in all the somatic cells. Somatic cells of the body means the cells of the body which do not give birth to the new generation.
Yes,
New
sir.
generation means it can't give a birth to the complete new organism.
Yes, sir. Okay, sir.
For example, in case of just I discussed with like for our body, if we discuss for our body, so all the cells of our body are not capable of regenerating the complete organ. It is only the sperm cells and the ovum which is capable of reproduction. So only when a sperm cell combines with an ovum, then only there will be a formation of a new organ. Otherwise, if suppose a, a skin cell combines with another skin cell or just one skin cell, will not be able to give birth to the new generation. Got it? So this is what we are observing everywhere in our body. So everywhere, what is occurring is metaphase. What is occurring is the mitosis cell division. Okay. You can see the details of metaphase are given here, where you have the appearance of the spindle fibers and their attachment with the chromosomes. In the anaphase here, you have the chromosomes being pulled apart. Okay, from the centromere, the chromosomes have been pulled apart. Got it? Then in the telophase, okay, so everyone understood about all the phases of cell division.
Yes, sir. Sir, is interface known as anaphase?
No, interface is not the anaphase. See, interface is the part of the life cycle of a cell. See, here we are only interested with the life of the cell where division is involved. Throughout its life, the division is not involved.
Yes, sir.
So when the division is starting, so that is the part. Division starts from the prophase.
Okay, sir.
Okay. Before that, it's the general life cycle of a cell. So that we call as the interface. Interface is the time or the life of the cell, between when the daughter cells are formed. So now the daughter cells will grow, they will
Sir,
develop.
yes, sir. Sir, is it possible that a cell cannot go through any one of these phases?
Cell cannot go through any one of these phases. So obviously, if it's not dividing,
Yes, sir. Okay, sir.
so if the cell doesn't divide, so it is possible.
Yes, sir. Sir, is this only for plant animal cells?
No, it occurs for the plant cell also. Just we discussed that in the animal cell there will be appearance of the spindle rays from the centrioles, whereas for the plant cell it will appear without the centrioles. They are called as spindle fibers.
Yes, sir.
Got it? Now let's see, as I discussed that I will be also discussing the content of motion, since Rohit has doubt from motion. Okay, so all of you also look for the graph discussion. So now we shall discuss the graph.
Sir, graph questions will only come for 3 marks, right?
How much?
So 3, because if they ask us to make a graph, will it only come for 3 marks?
See, from the graph, there are different types of questions that can be asked.
No,
Okay,
sir,
a
like if
number
we
of
ask
questions.
us to draw a graph for the given information, will that only be for 3 marks?
Yeah, that can be for 3 marks.
Okay, sir.
Okay, so from the graph, see what all information or what all different types of graph that you can get.
Sir, we
See,
can get distance time graph,
mm,
velocity time graph,
yeah,
speed time, and position time graph.
okay. So what all information can be asked to find out from them?
So they can give us the distance,
Mm,
then they can ask us to find the time if
mm,
they give us a speed, or they can give us a speed and time and ask us to find the distance.
Okay, see. So if you have a distance time or a displacement time graph, consider this to be a displacement time graph. So for simplicity, we are all taking this to be the displacement time graph. Okay. So for all the displacement time graph, what all questions can be there?
So
Of
they can
this,
ask us to find the type of graph, like if it's accelerated motion, what type of motion can be asked, types of motion can be asked, sir, related to the graph.
okay. So first of all, you can be asked type of motion. You can be
Yeah.
asked the velocity from the graph.
So the time or the displacement or distance from the graph.
Yeah. So,
So the
okay.
first one is accelerated motion.
Which one is first one?
Yeah.
No, it's not accelerated motion. So see, the first one is a uniform motion.
Uniform motion. Second one is at rest.
Yeah. So the second one is at rest.
Third one is deceleration since it's coming
No,
downward.
no, it's negative velocity.
Negative. Oh, sir, is this velocity time?
This is displacement time graph. So it
Okay.
shows that the displacement is
Negative.
decreasing. So in the displacement shows negative velocity.
Sir, then will the fourth one be acceleration?
So here the displacement is not increasing.
Yes,
Which
sir.
one?
Sir, fourth one is acceleration, right?
Yeah, fourth one is the accelerated motion. This one is an accelerated motion. Okay, so from this graph, you can be asked the type of graph, or if you are given the graph something like this 1, 2, and 3, so you can be asked to find out the velocity from the graph.
Sir,
So
when
how
is
do,
this?
yeah, how can you find the velocity from the graphs?
Sir, we can use the slope.
Yeah, we will find out the slope of this graph. So here slope of the graph will give us the velocity, right?
So
Slope of,
slope will be y2 minus y1 by x2 minus x1, right?
yeah, so slope will be y2 minus
Sorry,
y1.
sir. It should be s2 minus s1, t2 minus t1, right?
Yeah, yeah, you can use that. Okay. So this is if you are talking in terms of coordinates, so you can write it y2 minus y1 over x2 minus x1. The second one can be your velocity time graph. See what all different questions can be there from the velocity time graph. So from the velocity time graph, you have okay, see. So you consider this to be a velocity time graph. Velocity time. Again, suppose the shapes of graph are still the same. So there
Yes.
will be different information that will be conveyed in each of the graph. Now the information changes. So again, if you have a graph something like this, let's say, okay, you have a graph like this. Suppose you have a graph over here like this. You have a graph here like this. and you have a non-linear graph, something like this.
Yes, sir.
So here you can have a non-linear graph like this. So now, what about this first graph? What type of motion is shown by this graph?
Sir, accelerated motion.
Yeah, so this one, okay, all of you, Jitendra, Shravan, all of you must answer. Okay, so this is your accelerated motion. This is accelerated motion. Shravan, what is the second graph showing?
Sir, can I answer?
Yes.
Sir, negative acceleration, no?
Yeah, so it shows negative acceleration.
Sir, can it also be called deceleration?
Yeah, so it shows negative acceleration, or you can also call it as deceleration. So or you can also call it as retardation.
Retardation, yes, sir. Sir, the next one is uniform motion.
Okay, so this one shows uniform motion as the velocity is not changing with respect to time.
Sir, and last one shows non-uniform because the velocity is increasing and the time is also increasing.
Okay, so here we can say acceleration is non-uniform. Okay, in the first two, you have a uniform acceleration, but here you have acceleration as non-uniform. Okay. You obtain non-uniform acceleration here. Got it?
Yes, sir, I understood.
Now see, the questions which are asked from this, another type of questions which are generally asked from the velocity time graph. So from the velocity time graph, suppose if you will be given a velocity time graph, something like this.
Sir, it says find the
Hmm.
it says
Find the
find the
acceleration
yeah.
and find the displacement. Okay. So these are the two possible things which are generally asked from these graphs. So if you consider
Sir,
this
they can
graph,
also ask whether it's a non-uniform graph or uniform graph, right?
okay, let's see.
Yes, sir.
Okay, so something like this, generally most of the questions, this is the most fundamental question which is asked. So you have your O, then you have A, B, and C. So you are suppose if you will be asked the velocity of the or it can be asked like acceleration during the portion OA of the graph. Just let me
Sir,
correct it.
portion A of the graph.
During the region OA of the graph. Let me
Sir, it
correct
will
it.
be 0 minus 4
Hmm.
minus 0 minus 1.
Okay. So that's what I'm correcting the graph. It's like
Oh,
this.
0 minus 2, sir, sorry.
Yeah, so it will be 0 minus 2.
So negative 4 by negative 2, which will become 2, sir.
Okay.
Acceleration is 2 meter per second.
Okay. So in the first case, your acceleration is 2. And then you have to find the acceleration for the next portion.
Sir, is it BC?
Yeah, it is BC.
Sir, so it will be sir, it will be 4 minus 0. 4 minus
Okay.
0
Yeah.
minus
Shravan,
by 5 minus 8.
Yes,
Shravan,
sir.
can you answer?
Yes, sir.
Okay. So answer the acceleration during the region OA, AB, and OA, AB, and BC.
Sir, OA is 2, right?
Okay, so during the region OA, its acceleration is 2. And what is the acceleration during the region AB?
AB 4.
During the region AB, okay, is the velocity changing? What is the initial velocity?
Here it's uniform motion because it's moving with equal distances in equal intervals of time.
Yes, it is moving with equal in distances. See, the velocity is constant. During the region AB, it has a constant velocity of 4 meter per second.
Yes, sir.
So velocity is not changing, hence acceleration is 0. During the region AB, the acceleration is 0. What is the acceleration during the region BC of the graph?
Sir, it is 4 minus 0 by 5 minus 8.
Okay, 4 minus 0 over 8 minus 5 or 5,
8
sorry,
minus
5
5.
minus
Sorry,
8.
sir. T2 minus T1. So it
Okay.
becomes 4 by 3.
So
which
it's
will be,
negative acceleration.
Yes, sir. It will be
Okay,
negative some decimal point. Whatever value we get for 4 by 3.
The next one you need to find the displacement,
Sir, it's the area under the
This
graph. So
object.
area of AB.
Total displacement.
Yeah. So it will be area under the graph. So
Yeah, it is the area under
of,
the graph.
yes, sir. Sir, area of AB, rectangle
It is the
ABCD.
area of
Rectangle ABCD.
not only rectangle, it's the trapezium. You have to obtain the complete area.
Right, sir. Trapezium ABCD.
OABC.
Sorry, sir. OABC. Sir, it will be half into OA. Sir, it will be half into height into A plus B, which are the sides. Sir, so it will be half into OA. Half into OA plus so it will be half into OA into A plus B, which is the sides. So OA
Okay,
plus
so
BC.
you need to make half into AB plus OC multiplied with height, which is 4. So AB is 5 minus 2, that is 3, and the total time is 8. So you get 1 upon 2 times 8 plus 3 multiplied with height, which is 4.
Yes, sir.
Okay, so bye all of you. See you again in next class.
Then if I have doubts, can I post it on the group?
Yeah, I don't think I'm there in the group. Doubts group.
Okay, sir. Yes,
Okay,
sir.
you can send.
Yes, sir. Thank you, sir. Bye, sir.
Bye.
Thank you, sir. Bye.
Okay, bye.